The most exciting thing I think that has happened in research in the last 2.5, 3 years has been the explosion of biotech in British Columbia, and in particular mRNA packaging technology, lipid nanoparticles, that was developed at UBC starting from about 40 years ago, but really coming into its own about 10 years ago, has led that enormous explosion. My name is Leonard Foster. I'm a professor of biochemistry and molecular biology here at UBC, and I'm also a member of the Michael Smith Laboratories. In the last couple of years, as some of the big pharma companies started developing urgently vaccines for COVID, they realized that this mRNA technology was really mature and ready for putting out into the public. But a critical component of that is being able to encapsulate those mRNA molecules using lipids. The goal of the first project is really to understand how to minimize the number of ingredients that need to go into these vaccines, and then doing that in a way so that the vaccines are the most efficacious that they can be. The other project is aimed at understanding, when a new variant is discovered, how that variant is probably interacting with our cells. And if we can understand what those differences are, we could either develop new vaccines or possibly develop different ways, different therapeutics that could be used to prevent those variants from causing disease. We couldn't reach these goals without that funding. The funding is for different kinds of instruments that are going in 4 different labs, and they allow us to ask questions about these viruses and the vaccines that in theory we could ask before, but we'd never be able to answer, and now we're going to be able to answer some of those things.